Hello everyone, and welcome to today's webinar, At the Edge of Genomics, Sequencing Single Viral Particles One at a Time. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by GenScript. To learn more, visit genscript.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Manuel Martinez-Garcia, Associate Professor of Microbiology at the University of Alicante, Spain. Dr. Martinez, you may now begin your presentation.
Perfect.
Yeah.
Okay.
Hello, everybody. So it's a pleasure to give this presentation. I want to thank GenScript for giving me the opportunity to spread out what we are doing in my lab. I think I can summarize with this title of this presentation, At the Edge of Genomics, Sequencing Single Viral Particles One at a Time. So my name is Manuel Martinez. I'm the head of the lab of single cell and virus genomics, and we are in Spain, here in Alicante. This tiny dot is a beautiful place to live. As you see here, beautiful beaches. And I think that one of the most important points is if I'm here presenting these results, what we are doing is because I have a nice people working hard in the lab, as you see here, so I want to thank all of them. And also I think it's important to, at the very beginning, say thanks to our funders from different parts of the world. So for my presentation, I want to first introduce what I would like to present or explain. First, I'm going to focus on trying to explain what is single viral genomics. Probably you never hear that. Probably you hear single cell genomics. Maybe it's more familiar for you, but probably the concept, the approach, or the methodology of single viral genomics is quite new for you. So I think the first part of my talk try to explain different parts of this methodology. And the second part of my talk is going to be science focused. So I'm going to explain basically different examples, quite brief, about what we are doing thanks to single viral genomics. So I think it's important to start with this thing. So you see here one single cell. So years ago, different people from different labs started a new field that is called single cell genomics. Single cell basically try to address different questions, biological questions, genetic, phenotypic, physiological questions from a single unit, single live unit, which is the cell, right? And with that technology, we basically have unveiled really amazing things from different fields. For example, in cancer, now we know different important aspects on how different single cells evolve in a single tumor, or for example, a really nice studies on the genetic mosaicisms in the biology of multicellular organisms in the brain, or for example, in embryology, how the different single cells evolve differently. So I think it's important that we realize that now single cell is kind of a standard and feasible technology, but probably not 10 years ago. 10 years ago or 15 were quite new. So this new field called single viral genomics starts, of course, with a single viral particle. So as you see here, for example, when you take a saliva sample, you can find millions of viruses in your saliva, normally bacteriophages, per milliliter. So what about if we can obtain the genetic information from just a single viral particle. Is it possible? Is it doable? Is it really complicated? And this is kind of what I'm going to address in this part of the presentation. I think the size matters. So in any technology or any methodology that you want to apply in science, it's important that you realize the size of your population, the size of your object that you want to study. In the case of, for example, a small bacterium like the one living in our mouth or in the gut or even in the Mediterranean Sea or any other oceans, there is only 1 to 2 femtograms of DNA. This is very, very small amount of DNA. But when you look at viral particles, even that amount of DNA is even lower, right? So then it's quite complicated at the very beginning to try to decipher the genetic information from just one viral particle. But it's not impossible, and that's what I'm trying to convince you. If you click, for example, and you type single-cell genomics, there are like 2 million of entries. If you click in Google Scholar, single biogenomics, you're only going to find, for example, like a few 200 papers, and it's growing a lot in the last 4 years. So it's a field that is probably exponentially growing. Basically, if I want to, or if I need to define what is single biogenomics, is the sequencing of the genetic information from just one individual viral particle. And how do we do it? How do we do that in the lab? So step by step. Is it possible to retrieve just the genome from one viral particle and sequence that? And the answer is yes. And this is basically the summary of the methodology. So you can find more details in our recent Nature Microbiology reviews that you see here in the left part of the slide. But basically the methodology we can summarize in these few steps. So we have the community, can be from different biological samples, gut, soil, blood, even marine samples, sediments, wherever. So then what we do in the lab is we add a fluorochrome, normally that stain the DNA. For example, can be cyber green, cyber gold, so very famous and very widely used in the lab. And then we wait until this fluorochrome gets into the capsules, and then the viral particles become fluorescently stained. Then normally we go to a flow cytometry in which you can detect the fluorescence particle, and then you can tell the equipment to put just one viral particle in one well, for example, in these multi-well plates. Then, for example, here, from just a complex community, you can sort like 300 viral particles individually, each one in each one of the wells. And then after that, normally what we have to do is we need to break this capsid and release the DNA into the well. And then after that, we pass to the whole genome amplification. It is obvious that we cannot directly sequence just a single molecule of DNA. So we need to make copies. So far, with the technology that we have, we need to make copies, and we use different methods, and one of those can be multiple displacement amplification. And from that amplified DNA, then you can go for PCR screening, you can hybridize with probes, you can directly sequence the DNA. So this is basically the different methodology, the different steps that we have. There are different ways to sort viral particles. Flow cytometry is one of the most famous one, but microfluidic and probably nanofluidics is very promising. And as you know, we are in the era of microfluidics, and probably in a few, I'm not sure, months or a year from now could be doable. So then I'm going to put one example. What you see here is this is bacteriophage P1 sample is infecting Escherichia coli. And then what you see here is each one of the dots is a viral particle of this bacteriophage P1 that we stained with cyber dye, and then you can visualize very well under the fluorescent microscopy. Then you can go with this sample, you can go to the flow cytometry, and then you can distinguish very well the population stained with this fluorochrome. What you see here, this gate in red color, labeled as P1, is the population that is stained with the fluorochrome. In the y-axis of this plot is fluorescence signal, so in this case green fluorescent signal, and in the x-axis is the size of the particle. So of course, when you detect or try to detect viral particle stained with a fluorochrome, they are going to be in this lower bottom part of the plot, okay, because they are small. And then after that, what we did in our lab was we decided to sort like several 100,000 of these viral particles, and then we inspected under the confocal microscopy. And as we expected, we detected this viral particle in our sample. Also, we did like a test, a blank. We say to the flow cytometry, sort the noise where viral particles are not supposed to be. I remember that they are in the gate P1 because they show fluorescent signal. So this noise gate probably is debris from the processes from the sample. And as you see here in the confocal image, we didn't see these small dots. Also, we use a super resolution fluorescent microscopy. As you know, it's a very really interesting technique that increases the resolution power of your instrument, of your equipment. And then basically we tell to the flow cytometry, just sort one viral particle, and then we scan it with this super resolution with a STED microscopy, and we confirmed that the equipment just put one single viral particle. So then we are ready to move on, and then we decided to, as you see here, this is one of the viral particles, one of the examples. We decided to go ahead and take this methodology and apply to different biological samples that I will explain later. Okay. So then after that, you need to, as I say, to break up and pop up the capsules. So how do we do it? So in this case, we use normally, basically we use different chemicals in order to break the capsules. So in this case, we are using a very simple buffer that is KOH with pH 14, that basically with this shock, we aim to break the capsules. Also, we combine with a thermal shock with liquid nitrogen. Just combining these two things, we can break up and release the DNA into these wells. So then we pass to the whole genome amplification. In our case, we have tested different type of methodologies. I'm not going to go into more details, but I'm going to be happy to take more questions at the end. But for example, I'm going to show the results from multiple displacement amplification. So in our case, we do real-time whole genome amplification. So using this methodology as the qPCR or real-time PCR, we can monitor in real time the number of viral particles that are leading for a positive whole genome amplification. I want to mention that there are in the market other type of whole genome methods, as you see here, that Malvac, Picoplex, and many others. So in our case, with real-time multiple displacement amplification, you can see in real time, in just 2-3 hours, if your viral particles, your genome release, are being amplified as you see here. So each one of the curves that you see here, indeed, are coming from the whole genome amplification from these viral particles. So when you see a flat green line, it's because this viral particle is not amplifying anything, because of different reasons that we can discuss later. I think it's important also to mention that we need to put negatives because this technique is very sensitive to contamination. What you see in column 1 and column 24 are negatives control, and then you expect a flat line, which indicates that your reagents are good and not contaminated with external DNA. So then you have this kind of results in which you know very well the position in the plate that render whole genome amplification, and then you can go to the next step, whole sequencing, or different according to your purposes. So here also you can run in an electrophoresis the MDA, or multiple displacement amplification, or whole genome amplification product. As you see here, a very thick band of material that have been amplified. With that, you have really a lot of DNA for different purposes in molecular biology. So basically, from just one viral particle, you can get its genome amplified and ready to sequence. There are different ways to amplify this 0.2, for example, femtograms of DNA that we contain in a viral particle, up to micrograms of DNA in one well. You can sequence this DNA by using Illumina, PacBio, or Nanopore. In fact, we have tested all three technologies, and each one has its own advantages. I'm not going to discuss and go for more details, but this MDA, or whole genome product, is compatible with all three and probably 4 platforms that we have, including mutorrent amplification, sequencing, sorry. Also, we tested different assemblers, and then you can reconstruct, of course, and rebuild and assembly the genome of the original viral particle. And then, of course, there are pros and cons. I mean, there is no perfect technology, and I think it's important at this point that we dedicate like a couple of minutes to explain the benefits, the pros and cons of this technology. I'm going to start first with the cons. So far in our lab, we have tried whole genome amplification for DNA viruses. We have tried hard for RNA, but I have to say and admit that we have not a methodology, an enzyme to amplify just a single RNA viral copy. So I think it's important to mention that, because for those that are working on RNA viruses, and maybe if they want to apply this technology, we need to go and try to find new enzymes to amplify just one single genome copy from RNA viral particle. So we do not have the sensitivity yet. Also, I think it's important to mention that when you work with single viral genomics, basically you destroy, of course, your original viral particle. So no longer can be used for any other phenotypical or proteomic or whatever other idea you have in mind. So this viral particle is no longer existing. You, of course, have the genome, but not the rest of the components. I think it's important because it depends on your question, scientific question, which is what it really matters in science. I think it's important to mention that. But also, I have to say that with synthetic biology, you might overcome that. In fact, we are now conducting a research in the lab, doing, thanks to GenScript, to synthesize in vitro the viral genome from one of the viral particles that we recover in our research. And then we can clone this genome in, for example, a rescue system, and try to produce new viral particles. So again, synthetic biology also can help you on these questions. What about the pros and the benefits? Well, I think one of the important points is, for example, in microbiology, we know now for a while that most of the viruses living in nature cannot be grown in the lab. So there are several papers, and it's very well known in microbiology that most of the bacteria and microbes cannot be isolated in pure culture in our labs in a petri dish. So then, of course, the virus is infecting these microbes. If you cannot grow the host, then it's going to be impossible to isolate the viral particle infecting this host. So a way to overcome this is using single virus genomics, because you directly take the biological sample, you stain the virus, and then you can go and obtain the genome from these viruses, regardless you can culture or not, or you know the host or not. I think this is important. I think also in the second point, probably I already explained it, but you don't need to have the host. So in fact, you can identify and sequence the genome without having the host of the viruses. This is really great because it's opening new avenues in virology. Any DNA viruses can be virtually targeted by this technology, regardless the size. It can be big or small. So it can be targeted by single virus genomics. Also, the third or fourth point here is we can target, regardless the diversity present in the sample, any kind of viruses. And I'm going to explain this point more in detail later. So believe me, I'm going to hold on this question about microdiversity. And another important point is we think that metagenomics can recover the whole diversity, genome diversity in a sample. Now we know, as we demonstrated with single virus, that this technology is not perfect. And single virus genomics can complement very well other molecular biology techniques. So, and then I think it's the time to explain with a couple of examples the usefulness of this technology. So, so far we have used this technology to different samples. I'm going to focus in just a human microbiome example, well, in our case, human virome example, and also virology example contextualized in our oceans. I'm going to start first from this Mediterranean survey that we conducted in our lab, that is titled Uncovering Hidden Marine Viruses, in which we discover which is probably the most abundant marine virus in Earth. And I'm going to repeat what I'm saying. So until probably 3 years ago, we didn't know, and we were probably overlooking one of the most abundant viruses in our planet. And we discovered that virus with this technology. We thought by that time, in 2017, that we already have discovered the whole diversity of viruses present in our oceans. As we demonstrated in our paper here in the Nature Communication, we were forgetting very important pieces in nature. So, and then what we did was we took samples from different parts of the oceans, in the Atlantic Ocean and in the Mediterranean Sea, and we sorted several thousand of single viral particles. And we put just one single viral particle per well. Applying this technology, we amplify the genome, and then we sequence at random totally 44 single viral particles. So I'm not going to explain the results on this slide, but this is a whole genome comparison of 5,000 reference and representative genomes living in our seas, in our oceans. And what we obtained was that the 44 single viruses, the 44 viruses that we recovered with this new technology were new species. But not only new, also new genus, and probably some of them new families. I'm going to focus on the one that you see here labeled as 37F6. This virus that one of my PhD students wanted to call 007, like James Bond. I think I should do it, but anyway, it's not so sexy now, the name. 37F6 is the most abundant virus in our oceans, and probably one of the most abundant viruses in our planet. And we discovered that after comparing the abundance of this virus against the rest of the viruses that were in the databases in the NCBIs, in the gene bank. In the red color that you see here is the abundance, the relative abundance of our viruses in different parts of the oceans of our planet. And as you can see here in the bar chart plot, when we calculated the abundance for each one of the viruses, the top one was the 37F6 virus recovered by single virus genomics and was present in any single marine sample from any single location that we have studied so far. But the most incredible point was never we realized that this virus was present and living and inhabiting our oceans. And the main reason was because this viral population is super micro-diverse. And by using a standard metagenomic assembly and a standard genomic assembler, this struggle to recompose and reassemble this genome. And always we were missing these important pieces in our viral databases. So a single virus genomics can overcome the problem of microdiversity, the problem of reconstructing these super complex puzzles, just going from one viral particle at a time, we can overcome this type of problems of genomic assembly. With that, I think I can convince you that single virus genomics is a novel technology that can be powerful to unveil important pieces that we are missing in our planet. And the second example, I'm going to be brief, is we also try to implement the technology in the human microbiome surveys. And for that case, what we did was we took saliva from a healthy group and also saliva from people immunocompromised, in this case with immunodeficiency in IgA, which is what the most abundant antibody in our mucus. And I think it's, you know, that is important, and because it plays a role in defense against different pathogens. So we took the different samples and then we process through different, like standard metagenomics and also single virus genomics. So in this case, we recover several viruses. We sequence several viral particles coming from different viral samples. And as you see here, we unveiled the genome from different bacteriophages, in this case infecting Streptococcus, which is a very abundant microbe in our mouth. And when we plot the abundance, again, these viruses were ubiquitous, widespread between different samples, and very abundant in saliva. So again, with this technology, I think we can answer different fundamental biological questions in biology. I think we have, with this tour that that we have done in this presentation, we have covered what is single virus genomics from the very beginning to the very end, what is the usefulness and different application of single virus genomics. And with that, I think, again, I want to thank especially Oscar Fornas, which is one of my favorite collaborators, because he opened his flow cytometry facility. He's very crazy like me and very supportive, and we decided to try to implement single virus genomics in collaboration. So also I want to thank many other people from different institutions in Spain and abroad, Bigelow Laboratory, Dr. Joaquin Martinez, and Silvia Cinas, Pep Gasol, and other people providing samples, and my funders. So I want to thank for being in the other part of the lane, and I'm going to be super happy to take your questions and answer in the chat, or maybe later through email or whatever other things that we have for exchanging opinion. So thank you very much. And with that, I think I'm done. Thank you very much.
Thank you, Dr. Martinez, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, let's see, RNA is very fragile, in quotes. Is this one reason as to why it cannot be sequenced in this manner?
Okay, thank you very much for the question. I think it's interesting. So it could be a reason why RNA is very recalcitrant for amplification, but I think probably the important point is that we don't have an enzyme in the market with the minimum or enough sensitivity to amplify just a single RNA molecule, as we do for DNA. So, for example, we had a project, an ongoing project in our lab in collaboration with another lab in the United States to implement single biogenomics for RNA. And we have tested a bunch of commercial enzymes to be used in the lab, and they were unsuccessful. So that's what I can tell.
Thank you, Dr. Martinez. Our next question is, how do you pick up rare mutations in this technique if you are looking at one single viral particle?
I think it's obviously a very interesting question. So it is true. So if you only have a single genome, it's impossible to identify if you have a polymorphisms, an SNP, or a mutation. But in general, I think what I recommend is, of course, to combine, for example, with metagenomics, or if you have other reference genomes similar to your virus, and then you can compare your genome against a reference genome either in your in-house database or in public database. Also, another important point is maybe you can sort several dozens of viruses belonging to the same population, and then after sequencing you can compare all of them in order to check if you have some SNPs. In our case, what we did was, after sequencing a bunch of single viruses, we compare against several metagenomes coming from the same sample, and then we could identify the SNPs. I think with that, I answer more or less the questions. I think it's really interesting.
All right, thank you, Dr. Martinez. Our next question is, let's see, how can this affect, or can it be applied to the genomic sequencing of SARS-CoV-2 variants?
Well, so far we can't. Unfortunately, as I said in the first question, we don't have an enzyme with the sensitivity to amplify just a single viral particles. So you have to realize that this amplification has to be robust, has to be reproducible, and unfortunately we don't have an enzyme with such capabilities in the lab. There are like a few options that are not commercial, but they are on the way. So.
All right, thanks again, Dr. Martinez. We have some more questions coming in. Let's see. What is the most common issue seen in the technology/instruments used when attempting to sort through single viral particles?
So one of the things is, so I always recommend if to do the sorting in a core facility that has experience or a long trajectory working with small things, so bacteria or viruses, because many or several core facilities only work with eukaryotes. Eukaryote is a big thing compared to a virus, and then the scale is totally different, and normally they have the instrument more tuned or optimized to this big size. So my recommendation, personal recommendation, is to try to start with a core facility center with a long experience with this. If you go to one of these places, probably the issues, the typical issues that you can have probably is going to be, I would say, a piece of cake, because if the people, the director or the technician, they have a long experience detecting viruses and bacteria, it's going to be easier to do the sorting.
Okay, thanks again, Dr. Martinez. Our next question is, how do you make sure the flow cytometric reagent / consumables or buffer is free of virus?
Okay, so normally the flow cytometric reagents are sterilized, and also most of the sorters, flow cytometry sorters, have several filters in the fluidic lane. So in our case, what we do is we prepare in-house the reagents, but you can buy. So these reagents and seed fluid can be perfectly buy from different companies. In our case, we heat the buffer up to 400 degrees. So, and then we make this buffer and reagents very sterile, right? And also we filter through 0.02. But as I will say, they are sterilized, so viruses should not be present in these buffers and reagents used for flow cytometry sorting.
Thank you, Dr. Martinez. Our next question is, what is the error rate in the amplification step? Is it low enough to evaluate single mutations in the sequence DNA with certainty?
The answer is yes, because for example, the polymerase that we use in the lab and other people use for single-cell genomics is normally Phe29, has a lower error rate than most or all Taqman polymerases that are in the market. So Phe29 polymerase is a very vital enzyme, so you can trust, because if you trust in your Taqman polymerase during your PCR, then you should trust much more during the whole genome amplification, because the chance to introduce an artifact and mutation that is not actually present in nature is very low.
Thank you, Dr. Martinez. Our next question is, with regard to the challenge of analyzing RNA viral genomes, would it be possible to do reverse transcription of the RNA viral genome and then amplify the DNA genome that is generated?
Obviously, yes, and this is what we try. So we try in the lab, so first a retrotranscription and then an amplification, but one of the problems is, I think here the limiting, the bottleneck is exactly the retrotranscription. So normally we do retrotranscription in the lab in a bulk of RNA, so you have millions. So in a regular extraction from just a single cell, you have thousands of different RNA molecules, right? So in this case, you should think that we only have one. So when we try this, when we scale down until only a few molecules, we realized the amplification, the retrotranscription was not robust, was not reproducible. So then finally, our results indicate that we don't have an enzyme that can do as good as we do for the DNA. But obviously, yeah, the answer is this is the obvious thing to try, right? Retrotranscription, and then the second step, whole genome amplification of this cDNA. And in fact, the second step is not the bottleneck, because we can use the Phe29 to amplify this cDNA. So the complicated thing is to have this first step done with just a single molecule, you know, without introducing contamination, and also by using a robust technology that you can reproduce in any single lab. But so far, we cannot do it.
Thank you again, Dr. Martinez. Our next question is it possible to separate bacteriophages and bacteria and then sequence both with the single virus / bacterium sequencing?
Of course, I think it's a good question. Yes, you don't need to, you can have a mix of virus and bacteria in the same sample, and then what you can do is distinguish that in the flow cytometry, because bacteria, any single bacterial cell is going to have a higher size, right? And then with the flow cytometry, it's going to pop up in a different part of the graphic, separated from the viral population. So then you can tell to the flow cytometry, pick up, for example, a few cells or pick up a few viruses from the same sample. You don't need to separate and sort it out first by filtering or other centrifugation methods. You don't need to separate viruses from a bacterial fraction. So you can use the same sample, and then you can tell the flow cytometry to sort whatever you want. So in fact, we sometimes do that.
Thank you again, Dr. Martinez. Our next question is, can we amplify RNA?
No, as I said, from a single molecule, the answer is not, unfortunately. So we can amplify RNA from a few molecules, a few maybe RNA viruses, but not from a single level viral particle, RNA viral particle.
Okay, thank you again, Dr. Martinez. Our next question is, what type of specific equipment do we need to implement single virus genomics?
Okay, so we need I would say a couple of free instruments. One, of course, is a flow cytometry sorter, but I would not recommend to purchase one for your lab specifically. You can use one sorter that probably is in your university or research center. So there are many universities having this core facility flow cytometry centers. So the second instrument that probably you need to buy for your lab is for doing the whole genome amplification. So, but it's not very expensive. We are talking about 15,000 euros or dollars or 10,000. So it's not that expensive. This equipment I think is useful to amplify doing this whole genome amplification. And maybe you need to, if you want to do this whole genome amplification in real time, monitoring, as I show in the presentation, monitoring in real time, maybe you need to spend a little bit more, but it's not that much. It's 20,000 euros or dollars. And the other thing that is very, very cheap is a UV strata linker. So this UV strata linker is 5,000 euros. So other than that, the rest of things that you need is common for any single lab. Of course, a dedicated area for free of contamination with a flow hood specifically dedicated for this application. But other than that, this is what we need.
Looks like we have time for one last question. During whole genome amplification, I think it is important to consider DNA contamination from reagents or manipulation. How can we prevent that?
Okay, I think this point is important because DNA contamination can introduce a lot of biases in your data. So I think it's important to work on this. One important point is during the sorting, what we do is we use, well, now with the COVID, we are used to that, but we cover with a mask, with a glasses, with a lab coat. After sorting, what we do is during whole genome amplification, we use a dedicated specific flow hood only used for whole genome amplification. And another important point is to decontaminate as much as possible your reagents. And for that, you can do two things. Either buy and purchasing one kit that is in the market that already implement decontamination of DNA from the reagents, is served by QIAGEN. I have to say that I don't have any stock holds, anything from QIAGEN. And another thing that you can do is what we do in our lab, and I know that many other people working on single-cell genomics do, is They prepare their own cocktail, as many people do, for example, for a regular PCR. And then what we do is, when we have this buffer, this cocktail for whole genome amplification, we decontaminate it using ultraviolet light for a few minutes. With this UV, what you do is you cross-link the DNA present in reagents while you maintain the efficiency and the activity of the enzyme. The Phe29 enzyme is pretty robust. Even when you UV for a lot of minutes, still they work fine. The enzyme works fine, so don't worry about that. So, and with that, just doing irradiation with UV, you can remove and get rid of the DNA.
Thank you, Dr. Martinez. Do you have any final comments for our audience?
Yeah, so I think, so say thank to the audience. So I appreciate that they are in the other part of the lane. So I think it has been funny, and I appreciate your interest on this talk. And I also want to thank GenScript for organizing all of this, that I think is amazing. And thanks to all of you.
Well, thank you again, Dr. Martinez, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, GenScript, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Bye-bye.